Hello, everybody. This is our lecture on plankton and plankton communities. When we think about the plankton, we should think about we need to define it first. Okay, so there's these variety of words: ceston, plankton, necton, and benthos. Okay, so first off, we can kind of break things into the ceston and the benthos. Okay, so the ceston is all organic matter that's like suspended up in the water. So, take this picture over here. We've got all the plankton, the fish swimming around, but not the benthos. The benthos are those things like a crab here, or basically something that stays, kind of in the sediments. Um, you can take that ceston and split it into the plankton and the necton. Okay, now plankton basically can't swim. Okay, or really, if water is moving, they won't be able to swim against it. Whereas the necton are things like this fish here, this little trout here, or salmon here, can swim. And it can swim independently of the currents. If it might be swept away by a super big current, but it has the ability to swim. Okay, so we're going to be talking about the plankton today. Now, plankton are, there's so many different things. It's really hard to just narrow it down to one specific group because there are so many groups. But we can start to split it. There's a variety of ways to split the plankton, but we can start by thinking about phytoplankton versus zooplankton. Now, phytoplankton are those photosynthesizing plankton, whether we're talking about algae, bacteria, diatoms, dinoflagellates. There's all sorts of different organisms that you could consider within the phytoplankton. Then the zooplankton are animal-like. Now, there's certainly, like this little dinoflagellate up here has a flagella and is moving around, and its cell is moving around. So it's not, super. So it's animal-like in some ways, but it does have chloroplasts right here that are going to be like photosynthesizing. So some of these groups can kind of cross between the phytoplankton and the zooplankton, but certainly when we're thinking about like fish larvae, insects, rotifers, those are things that would be definitely within the zooplankton. So you might ask the question, well, what is the most abundant thing in the plankton to start off with? And the answer to that is viruses. They might not think of viruses as being alive. Some biologists do consider them alive. Most biologists consider them not alive. But this, 10 to the 8th, 10 to the 9th, a billion viruses per milliliter. Okay, think about that, milliliter. There's a billion viruses in there. The thing is, these are not viruses that are going to hurt you. These are mostly bacteriophages. So viruses that are going to be attacking cyanobacteria, heterotrophic bacteria in the water, or algae. Okay. The thing is, these viruses do a lot of nutrient recycling. Basically, if we didn't have viruses, nutrients would be locked up in currently growing algae and bacteria, and those viruses lyse those cells and really allow nutrients to get back into the water column. They really reduce the primary production by killing off a lot of the algae. They do release those nutrients, but then they're also reducing the primary production. So, but viruses are actually really, really important and crazily understudied. Then we can, kind of like take a step up in size, and we're thinking about heterotrophic bacteria. Those are those bacteria that are not doing photosynthesis, right? And these are insanely common also, about 100 million per liter. And, these bacteria are floating out through the water, absorbing those dissolved nutrients. Now, remember, a lot of those nutrients are released by viruses. If 80% of the primary production, if primary production is reduced by 80%, that's going to be, a lot of nutrients being released that can be easily sucked up by these bacteria that are extremely small. Really this viral shunt here, the connection between these two should be like super huge. Like there should be an arrow that's like super huge going this way and that way. So, this is certainly, while there might not be a lot of biomass in the viruses, there is going to be a whole lot of nutrient flow back and forth between these. All right, let's get into the cyanobacteria. Sometimes they've been called blue-green algae because they have only chlorophyll A, which gives them a little blue tint. You can see in some of the oscillatoria there, it has a little blue-green tint there. So these are the photosynthetic bacteria, basically. Okay, they look like a bacteria. They're structured. They're prokaryotes, but they're going to photosynthesize pretty much like a plant does. And there's all sorts of different forms. You can have, just single cells floating out in the water, filaments, or colonies, all sorts of different types. We'll talk about the succession of cyanobacteria and green algae and diatoms here in a minute, but we'll wait for that here for a second. So green algae, these are the algae that are in the chlorophyta group. These are the most plant-like of the algae. So we think that terrestrial plants evolved from this group here. They're mostly freshwater, but there are some, like all of these species right here are from marine systems. But and if you look at most of your food algae, so they're going to come from um, the oceanic green algae. This ulva here is highly nutritious green algae that you can eat. It's called sea lettuce. But it's also very, very nutritious for zooplankton and stuff. So they're going to be eating these up like crazy, and actually humans can survive by just eating green algae if they really wanted to. So these are commonly in midsummer in lakes in a classically dialectic lake. So the third really important group are diatoms. They're in the group Bacillariophyceae. Bacillariophyceae. And they have essentially glass cell walls. So it's made out of silica. That's what glass is made out of. And they're really pretty, actually. So this is an example of one right here. Those glass cell walls are, at this scale, at this size, glass is not, super brittle. That actually makes a pretty nice um, a strong, sturdy shell. These down here are not just a single algae, but it's basically individual diatoms arranged on a microscope slide to make these pretty cool designs that are oftentimes common in like Victorian Europe. Rich people would commission these art microscope slides, and then they would show them, show their friends these microscope slides of these pretty designs. Anyway, whether we're talking about a diatom that looks like this is Asterianella, or something more like classically like this or this one, they all have basically a frustrule. That's their cell wall that looks like a petri dish, and I think you can see that very easily here, where you have, the outside here of the dish, and then this one like fits inside it. So if you look at that at a cross section, it essentially kind of looks, like that, okay? Like a petri dish, right? So what happens is when they reproduce, sorry, that's supposed to be an arrow here. They're asexually reproducing. So and what you get is it turns into two, and then they grow another frustule on the inside. Now this one, you notice, is exactly the same size, or if had I drawn it correctly, number 1 and number 2 should be the same size. But notice now number 3 got smaller. So if number 3 were to reproduce, we would have one that was, this big, then another one. Let's say I drew that the same size. So 3 and 4 would be the same size, but this 5 now would be smaller. So what they need to do is essentially, if 5 reproduces, it's going to be way too small. So what they do is sexually reproduce, and then that brings them back to growing at that size, the original size. Okay. What we see is that also that diatoms are common in the phytoplankton, right? There's plenty of free-floating diatoms, but there's also extremely probably the biggest portion of the periphyton, the encrusting algae. Okay, but again, there's lots of free-floating forms. Now, there's all sorts of different other algae that I don't want to talk about, but all of these are minor players. It depends. I mean, depends on where you are in the ocean. Red algae can play a huge role in the ecology of the system. Brown algae are kelp over here, and they can play a huge role in the system. But when we're thinking about lakes, it's really three main players: diatoms, green algae, and cyanobacteria. So let's move into the zooplankton. The smallest zooplankton would be the protozoa. Three main groups are the flagellates, the ciliates, and the sarcodines. Usually what we think of as Euglena, Paramecium, and amoebas. Now, again, a lot of these things are sometimes photosynthetic, so they overlap with the phytoplankton. Where we find these in lakes are oftentimes in the upper hypolimnia. Okay, so relatively deep in the lake, and they can be moving up and down very often. They're not going to be able to defend themselves very easily because they're so tiny, but so they do this deep vertical migration that is pretty cool. Where they're at nighttime they're down. At least the photosynthetic one, like the photosynthetic euglena and some of the dinoflagellates, will be at nighttime be in the hypolimnia, or sorry, they'll be in it's a kind of a reverse vertical migration. So at nighttime they'll be deep getting nutrients, and then they'll come up for sunlight a little bit to get during the day. And that's different than what we think about for the vertical migration of zooplankton. One step bigger than are the rotifers. Now, most rotifers don't move around, but there's plenty of ones that do swim freely throughout the water column. There's only 2 marine species, but most of these are freshwater. And they're called wheel animals, or they have a corona, which is called. And they have, you can see it here, where right here there's like a little row of cilia. And what those cilia are doing, they're completely beating in. And I think you can see it here, where the algae in this little GIF here, the algae are running, being pulled due to those cilia. You can see the little cilia right in movement, but right in that area. What you can see is that there's a continuous flow of water going into this, basically the mouth of this animal. And if you look right here, then you can see there's some like internal teeth that are grinding up those algae and eating them. So this corona is used as for locomotion and feeding. It's a kind of multi-purpose organ, and it's pretty crazy. And around plants, these rotifers can be extremely high densities, 25,000 per liter. But usually out in the deeper water column, I shouldn't necessarily say deeper, but out in free floating in the water column, there's only about 1,000 per liter. But still, that's quite a lot. These are some types that we oftentimes see around in lakes here in Wisconsin. This is Asplankna. This is Caratella. These two are 2 different species of Caratella. And then I forget what this one is, but there's a whole bunch of rotifers that will look like this, and this is Polyarthra. This should be an O, sorry. So there's these look like have some pretty cool little wing structures, but they actually they don't swim with those things. Just kind of directs them in the water and keeps them floating. But so there's all sorts of different shapes. I would say that this shape is like the most common, kind of your general rotifer that has the mouth or the teeth there, then the corona up here. And sometimes I don't know why I'm continuing to draw. Sorry. Sometimes the foot here will attach to things and it can stay around for a little bit. It can like attach to a plant or a rock and it can stay there without having to swim around. But the most important part of the zooplankton are the crustacean zooplankton. There's two really main groups that we think about here, the Clubhasera and the copepods. These are the most common types of zooplankton by biomass. Their numbers might actually be lower, but they'll be by biomass. Sorry, their numbers will be lower than rotifers, but because they're bigger, there's more biomass of these things. So this is the most important link between basically the phytoplankton and then the pelagic, the fishes and all the metazoans, the bigger stuff that's going to be in the lake. So cladocerans are an order within the brachiopods and then within the phylum arthropods. So both of these are arthropods, but Cladocera in the class Brachiopoda and the copepods in the class Maxillopoda. So Cladocera, they can be really differently sized. There's like all sorts of different species. Some can be this Leptodora is actually can be 2 centimeters long, and I've got a short video that we can show here. Let me get out of this here for a second. And oops, went to the end of the video. Sorry. There we go. And this is a scuba diver diving. You can actually see these things with a naked eye. They're very transparent, but they can be almost an inch long. So that this guy is very close to scuba diving with it. But yeah, it's a really cool predatory kind of zooplankton that will actually eat these other brachiopods, like a Daphnia here or a Bosmina here. And their body kind of looks kind of like there's two shells. In certain views you can see it, but it's a transparent shell that's right here basically, and it kind of like think of if a crab were to like back. So the clamacerans are looking kind of like a crab that backs up into a mussel shell, and that's what their those mussel shells are like, really very thin, so they're transparent, but it protects them pretty well. So they have pretty much every species has a large compound eye, and they swim with their antenna. Their antenna kind of beat, and you can see that happening here. And what's interesting is when you watch them swim, they'll kind of like swim and stop, swim and stop, swim and stop, swim and stop. And they're swimming in that jerks because remember, they don't have any inertia. So as they're swimming, the friction from the water really holds them in tightly, and they'll not be able to coast because they don't have any inertia. Copepods, then, there's 3 types that we'll see. The calanoids, which are filter feeders. So this is a calanoid right here. And they're usually a little bit bigger. And what they'll be doing is filter feeding the water, and oftentimes their gut, which you can see right here, is filled with, it just looks green because it's filled with algae. The cyclopoids then are over here. And actually, this is a cycloploid up the GIF there. And the cycloploids then are they're actually a little bit smaller than the calanoids, but they are predators. So they're going to be eating other copepods, and they're going to be eating other like Daphnia and Cladocerates. So oftentimes you'll see, I don't know if you can really see it here, it's kind of in the background here, but right there, there's an egg case, and then the cycloploid here has two egg cases. And they carry their eggs for a while and let them mature until they hatch. Then the harpactocoids. These aren't really planktonic. They're really littoral scrapers that live within the sediments of like the edge of a shoreline of a not a stream, but a lake. And yeah, we don't really see them ever coming up into the water column. There's a whole variety, class of plankton really, that we don't really think of as plankton, but really, really are. Crayfish. Crayfish, this is a crayfish, basically larva, okay? Um, we think of crayfish definitely as benthic organisms, which they are, but they're meroplanktonic, meaning temporarily planktonic, um, as larvae right after they hatch out of their eggs. Fish, walleye and perch have planktonic eggs. The fish will live like their little larvae. You can see it right here. They're so tiny, and they have really no ability to swim when they're really, really young, and they just kind of go wherever the current takes them. We often don't really think of them as important in lakes. That's not true in oceans because there's plenty of times where people are really, think about the meroplankton more and spend some more time thinking about the ecology of them. In lakes, they're probably not as important, but for certain species like walleye, yellow perch, every crayfish, those meroplanktonic stages are super, super important for their species. So, in some ways, we should think of plankton as the most important part of a food web, right? Pretty much everything in the lake can be tied back to plankton. Most lakes are dominated by the plankton is the largest component of the biomass, just because most of the lake is, pelagic and not around the shoreline, and there's going to be the water might be pretty clear, but there's still a lot going on there, where the majority of the production in a lake is usually due to the plankton. So they really drive the base of that food web. So I want to spend some time thinking about why plankton size matters, because we can find very, very small plankton, right? Like single-celled, tiny little heterotrophic bacteria and Pychoplankton, to the super big plankton. This is a pyrosome. It's a really crazy. It's like this tube of planktonic creatures. They only live in the ocean, but they just kind of go where the current takes them. They're very rare to see. Usually they're down pretty deep. And then there's all sorts of species of jellyfishes that will really be planktonic. They'll just be floating away around and being taken wherever the current takes them. So there are some big plankton, like we just talked about, but really most of the plankton are pretty small. So when we think about for a photosynthetic plankton, size really is really important. So if you take the surface area of a sphere, it tells a lot about what's going on. Okay, so let's take a 4-meter diameter sphere. Okay, so we've got a sphere here and diameter 4 meters. Okay, the surface area then is going to be 50.3 meters squared, and the volume is going to be 33.5. So what this is saying is that there's, for every cubic meter there of volume, there is 1.5 square meters of surface area. So why are we talking about surface area? Surface area is really determining how much stuff it can pull in, right? So if you look, this 4 meters, let's just say this is 4 millimeters or whatever, for micrometers, doesn't really matter. But if we were to look like, on a little patch of this cell membrane, so we take that cell membrane and look at it, and what it's going to have is places in regular intervals, transport proteins and things that can suck up nutrients, okay? And there's only a certain number of transport proteins that you can put per area of cell membrane. So if you have a 4-meter diameter, a ball here, you can only have this. You only have 1.5 meters squared per cubic meter of that volume. Now, if you have a 1-meter diameter one, so we make a smaller ball, right? That's only 1 meter in diameter. Sorry, that probably should be smaller given than that. What you end up with is over 6 square meters of surface area per cubic meter of volume. Okay, and what that's saying is that, like, basically more membrane is feeding a smaller volume, so you're able to grow faster. And if you have 0.1 meters, you get a ratio of 59.2 to 1 for that surface area to volume ratio. So, as we get smaller and smaller and smaller, that number just explodes and gets much, much, much bigger. So why does that matter? Basically, what we see is the smaller you are as a plankton, the more efficient and productive you can be. There's faster biology for a variety of reasons. You can pull in more stuff to feed a smaller body. So it takes basically less time to grow to be the size that you need to be. That adds to more production, and then you also don't have to transport stuff around your body. If you're just a really tiny spherical algae, diffusion works fine, so it doesn't take a long time for nutrients to get around your body. So what we see is in oligotrophic lakes, we have really, really tiny plankton because it can be so efficient. However, in eutrophic lakes, you don't need to be efficient, right? You're bathing in nutrients. So the plankton themselves can actually be a lot larger. And it's good to be larger because the larger you are, the more you shade out things below you and you don't have to compete. And you can, the larger you are, the more sunlight you can grab. And in a dark eutrophic lake that's really murky and turbid, that's really important. So think about our plankton, right? Think about how an organism can survive, right? If you're a phytoplankton, sinking is a really big problem. Okay, once you go below the thermocline, if you don't have a way to swim, how are you ever going to get back up, right? And if you can't swim against the currents, basically you're just going to sink forever and die. So what we see is that some there's the oil droplets there. This is a diatom that has it using oil droplets to keep it more buoyant. This is an Asterianella diatom that is using projections and keeping its surface area really, really big so that it doesn't sink as fast. And then sometimes they do let the current resuspend them because, in the summer, the wind is going here and you do get a circulation here. So if they do start to go down, they'll get resuspended. As long as they don't sink below that thermocline, they'll get resuspended, as that wind mixes around the upland of that lake. So when we look at patterns of phytoplankton, though, might be what you would expect, right? At any kind of normal lake, you'd expect the most growth. So this is photosynthesis here on the x-axis, and this is depth from, this being the surface, and this being deep there. And what you're going to get then is this is, kind of what we'd expect, the maximum growth, because this is where the most light is going to be, right? And then as you go down deeper there, that light tapers off and you won't be able to have as much photosynthesis. However, in the most clear lakes, what we see is an inhibition, basically maybe in the first meter, there's too much UV light, the water is too clear, it might get too hot, and we see some photo inhibition. Basically, it's algae getting killed off because there's too much light. If you have a really eutrophic lake, what you'll generally see is, that there won't be any photo inhibition because basically there's a huge, really fast drop off of light because there's so much algae in the water that this biogenic turbidity, meaning murkiness due to things living in the water, right? It's basically just because you have so many plankton, you only have life up in the uppermost top layer because everything below that gets shaded out. Sometimes you get a metalimnion growth maximum, so this would be in a really oligotrophic lake. You might have some photoinhibition right there, and then the only place where basically what we would get here is kind of where the metalimnion is, and it kind of straddles that line between nutrients down here and then sunlight up top. So if you live in this metalimnion and kind of travel up to get sun, down to get nutrients, up to get sun, down to get nutrients, oftentimes you get a metalimnion growth maximum there. So when you think about the seasonal succession of phytoplankton, it's a really cool story where you get an increase in and so essentially the lake is frozen over, and then you get sometime in March, let's say, you get a real fast increase of diatoms, but the diatoms crash due to silica limiting them. They run out of silicon, so the green algae come up, and I shouldn't say they come up, but their populations explode and populations increase. And this is about the time when zooplankton are starting to be in really high numbers, and their populations then get decreased due to zooplankton eating them. Meanwhile, through all this time, then you get the blue-greens populations exploding because they do well in really warm water, and so July, August, September, the water is warm, and they outcompete the green algae. As it cools off, then, and say late September, October, the blue-greens don't do so well, and they start dying off. Meanwhile, silica has been able to increase and does well. The diatoms do well because they have enough silica to make their cell walls, and they do well until basically the water gets too cold somewhere in November, and maybe the lake starts to freeze over. So one common thing that we often see is that a lot of plankton are extremely clear. They're transparent because they're hiding in a completely transparent medium. So, but one other way that we don't really think of as a way to hide necessarily is hide via size. Okay, and what we see is that this paddlefish here has these gill rakers. If you look in between, like, so their gill is like right here and right here, but in between that gill, they have a bunch of bones that project into here, and you can see them right there, and they're called gill rakers. And what they are is essentially just a net. Okay, so the paddlefish opens its mouth and swims through the water, and the zooplankton get caught in those that gill raker net, and then they get swallowed. So if zooplankton is small enough, what it can essentially do is go through that net, and it can escape those filters. So whether it might escape a filter for like a mussel or this paddlefish or something like a gizzard shad. And then for another way to think about it is think if there's a tiny little zooplankton that this Leptodora here. Remember this Leptodora is about 2 centimeters long here. So it's a relatively big organism. If it's just trying to go after like really tiny particles, remember this is basically a hunter that's hunting through the water. If it's something that's so small, it's not going to want to go after it. It's not going to waste time, waste energy going after something that is so small and doesn't give them that much food to begin with. So another thing that organisms do, that the zooplankton are doing, are doing this thing called vertical migration. So this is a light-mediated circadian rhythm. And what we see is most common in the cladocerans. Copepods don't really do it all that often. I'm sure there's some copepod out there that does it. But zooplankton essentially come up at night, come up into the shallow water to feed. And what they're doing is they're basically hiding from those zooplanktivorous fish. Okay, because these things are pretty big. These might be like 0.5 millimeter from like here to here, and fish can see them. Those fish are those zooplanktivorous fish are going to be swimming through the water looking for these zooplankton, and especially they can see like the intestinal tract and the eye relatively well. But they're visual predators, so at night they don't do well, and so the zooplankton will come up at night, feed on all the phytoplankton, and then go back down during the day so they don't get eaten by all those fish. Phytoplankton then kind of do it the other way. They'll rise during the day to get sun and sink during the night to get those nutrients. Kind of talked about that when we were looking at that metaluminous growth maximum. All right, with that, I'll see you later.